Uh, okay, Sohela, would you introduce yourself? You're on mute, Sohela.
I'm also part of the Catalyst team. I work mainly on diagnostic projects or digital health projects that have a diagnostic application. I've been with the program about 2 years and really excited to kick off another year's worth of internship. Or summer's worth of internship, but another year.
Yeah, and thank you, Sohela. Nate?
Thanks, Sohaila. Thanks, Rupa. Yeah, it's my pleasure to be here. Welcome to all the interns. We are thrilled you're here. My name is Nate Prorock, and I've been with the CALS program for a few years now, and I'm working on mostly the digital health projects. And so I'm happy to facilitate those projects. And then another hat that I wear at UCSF is running the National Science Foundation I-Corps. And so for the next several weeks this summer, you all will be working on other people's innovations. You'll be working on these fantastic ideas from UCSF principal investigators. And I imagine that you all are here is because you're interested in innovation and by some extent also entrepreneurship. And so if you also have any ideas for your own personal innovations or entrepreneurial ideas, you'll be hearing from me more about National Science Foundation's I-Corps at UCSF. And what that entails is a series of customer discovery interviews over the course of no more than 2 weeks to ensure that your idea has traction, has customers. And so stay tuned for more on that. We hope to be having the next course sometime in the fall. Thank you very much and have a great Catalyst summer internship.
Okay, I will just orient you to the Catalyst program. So I will first do that. And I also want to thank Charles for sharing these slides with me. So the Catalyst Summer Internship Program, y'all are a vital part on working together to put the target product profiles. And I'm just, so the Catalyst program, we give seed funding in therapeutics, medical device diagnostic, digital health. And we have funding is up to about 100K. And we also have great industry advisors who are under CDA or confidentiality agreement, and they provide mentorship, they review applications, provide mentorship. And of course, all of you, the Catalyst interns, you all are grad students, undergrads, med students, postdoctoral fellows, clinical residents, and you work in teams along with the awardee, Catalyst awardees, as well as the Catalyst program director, and put together target product profiles. And I will introduce Charles later, and he will initiate what the target product profile is. And so our awards are in different categories. Like you may have therapeutics, you may have a small molecule, you may have a cell therapy, gene therapy, biologic, and similarly medical devices. They may go through a PMA pathway, where you'll have to do clinical studies, or you may have a predicate, go through the 510K pathway. And you will learn all these as you put together the TPP. Similarly, diagnostics, it may be a clinical, it may be a companion diagnostic as a test for treating people. Again, you may have to go through the FDA process, or it may be a lab developed test. And again, when you're developing the diagnostic TPP, you will look at these fields, learn more about that. And similarly with digital health, it may be an app, it may be clinical decision support. You learn about HIPAA compliance. And of course, now a lot of them have AI components in them. Now, this is just like there's so many different aspects in therapeutics development, diagnostic, medical device, or digital health. And this just gives you an overview of all the things that need to go in. And for example, in therapeutics, the Catalyst Award may be for preclinical studies or for hits, generating hits. And similarly, in medical device, it may be a prototype development or studies in animal models. There are so many different ones. And by the way, y'all will both get the slides and the recording. So if I'm going fast, you'll have time to look at the slides. And these are all broadly our advisors' expertise. It may be very specific, for example, in drug formulation or in chemistry, or you may have a venture capitalist who's looking at what is the potential of this to go forward, get funded, to get an exit acquired by a pharma company. And from Catalyst, projects they maybe go to an incubator, they may be licensed out, get additional funding. The idea is to move projects along the translational pathway. You start early and you want to obviously get it into a product for patients. That is the long-term goal. And this year we had 60 applications. They were reviewed by the industry advisors, 36 projects pitched as finalists, and 19 Catalyst awardees. And you all are all going to work in groups on the different projects. And you all are going to put together the target product profile. Nate already mentioned the regional NSF ICOR. Just some metrics. 313 projects since the inception, 13 million in funding was given to these projects, and a total of follow-on funding, 751 million, whether it is institutional funding, philanthropy, federal state grants, or like the startup company, for example, in Series A, may have raised 67 million, etc. And these are the companies spun out from UCSF, from Catalyst Technologies. And the ones circled are the more recent spin-offs. And among them, a few others are also recent spin-offs like Delve Bio and Air. These are the 19 projects, and y'all all got a list of projects and y'all picked your top 5 choices, and we tried to accommodate as close to your first, second, or third choice, typically. And Catalyst program is been earlier stage, and this is just example in therapeutics, 25 to 100K, while the Invent Fund has been typically giving 100 to 500K for a later stage projects. And these are the different topics, and hope to see you all at all of these sessions. They will be recorded and you will also get the recordings. And now let me stop share. It is my great pleasure to introduce Charles Hart. He was executive director of the Catalyst program for a very long time, a wise sage of the Catalyst program, mentor to the program, mentor to me, and really great to have Charles introduce the target product profile.
Okay, thank you very much, Rupa, for the introduction. Nice to see Nate and Sohaila and a couple other people on the call that I know, and they know who I'm talking about. So I myself am a molecular biologist. So I spent after my postdoc, I worked in biotech. I was always here in the Bay Area at venture capital backed startup companies. Although relevant to today, 2 different times, our little company, it wasn't mine, of course, but the company I was at was basically purchased by Big Pharma, by a large multinational pharmaceutical company. And so that's why I'm familiar very closely myself with target product profiles. And I'm going to say a little bit more about the specifics of what I'm alluding to in a minute. So almost like 7 years ago, I came to UCSF. I was hired to run the Catalyst program, as Rupa mentioned. And for the past couple of years, I've also been running UCSF's campus core facility focused on drug discovery. So UCSF has a little core facility focused on drug discovery. And just recently, I left Catalyst and I'm only doing the core facility. And so I'm gonna make a quick pitch that for any of you or your friends, whether you're UCSF or not, keep in the back of your mind that UCSF has a small molecule discovery center. And this could either be if you want to do some chemical biology experiments, complement genetics and biochemistry with chemical biology approaches to perturb biological systems, or you got a nice cell model or a new target and you want to, and you think it might be a good target for drug discovery or a good phenotype for drug discovery, don't hesitate to reach out to me and see, what sort of fit there is. And it's anyway, I don't have to say any more about that. So hold on a second. I had to click OK. I have a slate. Oh, here we go. No problem. So I'm going to say a few words just in general about translational research. So at UCSF, there's 3 times the research taking place. And this is true for, let's say, the world. There's basic research, as our chancellor likes to describe it, curiosity-based, fundamental discovery, basic research, just knowledge for the sake of knowledge. And at the other end, there's clinical research, asking questions, whether drugs work or not, whether medical devices works or not. And then in between is translational research. And I'm going to talk just a little bit about translational research. And then, as Rupa mentioned, I'm going to talk about the target product profile. And I'm going to home in on some of the fields that I'm familiar with because they have to do with drug discovery as opposed to medical devices or digital health that are going to be in subsequent weeks. And then if we get to it, we might not get to it. I have a couple slides on this concept of the entrepreneurial mindset that Nate and I like to present. It's just sort of for fun if we get to it. Okay, so here's the thing, and this says what I've already said, is that on the left side of this is basic fundamental curiosity-driven research. And I think you all know that UCSF gets more funding than any other public college, any other public university, and we're number 2 overall in America. And then on the other side is clinical research. And this is sometimes referred to as the bench to the bedside, taking things from the laboratory bench to the hospital bedside. And the phase in between the bench and the bedside is the translational phase. And it's sort of an awful term, but it's caught on. They use it at the FDA. They use it at the NIH. This paper, you can see at the bottom right, I took this cartoon out of Nature, the journal Nature. They refer to this translational phase as the Valley of Death. I said it was awful. It's called the Valley of Death. And you can see at the top left, there's the bench scientist, and he's got some cool discovery. And then you can see the physician, the healthcare provider at the top right, and she's got a patient that needs some help. And you can see that the idea died on the way, and you can look at the bottom there and you see the skeleton. And so, yeah, they call it the Valley of Death because it's where good ideas die because they don't get to the finish line. And it's pretty important, but given the current state of the world, there's lots of things that are important. Now, what is sort of underappreciated is that the Valley of Death, it's really mostly a money thing. It's not totally a money thing, but it's much a money thing. Because I already said is that on the right, there's plenty of money. Well, there was for basic fundamental discovery research. And if you look at the right, and for those of you that watch television at night, the number of commercials for drugs is unbelievable. And, you know, they're often for psoriasis or mental health issues. And we've seen them. For those of you that watch TV, I don't have to say anything more. So it just tells you that these pharma companies have tons of money. And it's not just when the company gets into the pharmacies and is on the market and they want to sell it and they want to convince you to go to your doctor and say, hey, I saw an ad on TV. Do you think I'm a candidate? Because that's obviously what those ads are for, because you have to get a prescription. So it's really, well, sort of, your doctor that that decides. Now, in addition to tons of money, once a drug is in the pharmacies, when a pharmaceutical company makes the decision, let's put this drug candidate into clinical trials, which is where the real money gets spent. I mean, the discovery phase, the preclinical phase, it can be expensive, but when you get to the clinical phase, it gets super expensive. And they want to be damn sure, they want to be as sure as they can that the molecule is going to be a success. And so it's really no holds barred, no expense spared. But this phase in between, there's not a lot of dough for. There's not a lot of money for. And if the next slide is not what I thought it was, let me just see something really quick here. No, I just completely messed up. Let me just use words. We're good with words. So here's the thing. I'll try to do this fast. If you're like me and you spent your career as a drug discoverer, and let's say you've got a nice target for, it's specifically for this rare form of high blood pressure, hypertension, that teenagers can get. And you're like, oh yeah, I've got this great drug. And you go to the NIH and say, hey, I've got this series of compounds that treat young adults or adolescents for hypertension. And what I really want to do is I want to do a series of tests to show that they're not mutagenic, carcinogenic, or teratogenic. That because the people taking this drug are so young, they have so many years of life ahead of them, you don't want any risk that the drug might increase cancer. Now, if you're an older person like me, and unfortunately, you have something like pancreas cancer, and I'm developing drugs for pancreatic cancer, you do not even ask the question. You don't even care if it's mutagenic, carcinogenic, or teratogenic, because the patient, unfortunately, is probably going to die within a year. And so they don't have 10 years left of their life to develop a blood cancer or 25 years left of their life to develop a solid tumor. But for a drug that's going into a kid or a teenager, it has to have a completely clean bill of slate. And when you go to the NIH and say, oh, I want a grant to do this, it's super important. You know what they're going to say? They're going to say, that's not creative. That's not anything interesting. All you're doing is checking boxes. No, we're not giving you a grant. And you're like, okay, cool, fine. So you go to a venture capitalist and say, hey, I've got this fantastic drug. I want to start a company. It's for teenagers with high blood pressure. And then the venture capitalist would go to you and go, hey, this looks really promising. There's not a lot of cases, but it's an important disease state. And oh, have you shown that your drug candidates are not mutagenic, carcinogenic, or teratogenic? And then you got to say, oh, no, that's what I need money for. And the venture capitalist will say, okay, look, I'm interested. But you got to check those boxes first. You got to do that work first, and you come to me. Another quick example. You have an idea for a drug for obesity. being a little overweight, and it's proteinaceous. It's peptidic based, and so you can't swallow it, because if you swallow it, your stomach will destroy it. So it has to be a shot. And I think you're kind of rolling your eyes. You're thinking, wait a minute, Ozempic's a shot. Yeah, Ozempic's a shot, but it's this little tiny baby needle that goes into your belly or goes into your thigh. You can barely, well, I'm not going to say that. You can, I was going to say you can barely feel it. I'm not going to say that. I've never tried it. I've never tried one of those baby needles, although my wife, it doesn't matter. She had to for a while, and she said it wasn't bad. But here's the thing. If the UCSF professor says, I got this great drug candidate for diabetes, and it's a protein, and lots of drugs get injected. There's lots of IV drugs, lots of subcutaneous drugs, lots of intramuscular drugs. And she goes to the venture capitalist, goes, hey, I got this great drug. And the venture capitalists say, no, it has to be oral. Doctors and patients want to take pills. They don't want shots. And so these questions you ask in the translational phase, does it cause cancer or not? Is it orally bioavailable or not? On one hand, they're not creative. They're just checking boxes. But on the other hand, I know you can tell this, they're super important, because they make the difference between whether the thing will be a success or it won't be a success, or the thing will be funded by a venture capitalist or it won't be funded by a venture capitalist, or it'll be licensed by a pharma company or it won't be licensed by a pharma company. And this is really introduction to the target product profile that tries to capture that. Hey, if anyone has any questions or this and that, I can't see
And
hands or this or that. So just signal that you have a question, and Nate will say, hey, Charles, there's a question from Brenda, or something like that. Okay. If you look at the left, this is a paper, this is a graph from a paper. This is a figure I grabbed out of this paper. I know, draw your attention to the fact that the y-axis is logarithmic. The y-axis, the vertical axis is logarithmic. These are papers published on translation between 2000 and 2013. And you can see, well, I had to point something out very important, very, very, very important. I already told you that I spent 30 years as sort of a translational biologist. The term translation is actually new. It didn't exist before the year 2000. And so I'm pointing that out because it's not like people didn't do preclinical research back in the 1970s or 80s or 90s, but they didn't use that word. So it's a bit of a red herring. But on the right is actually, I did this, and this is using the term translational research. I went into PubMed, and these are thousands. This is a linear scale, but it goes up in the thousands. And so you can see in 2008, there's 2,000 papers. In 2020, there's 15,000 papers. My point is this. My point is that the field of translation is sort of booming. And these are journals, and at least a handful of these are reputable journals. No, there's more than just a handful. There might be a couple of bogus journals here, but some of these are top drawer, like Science Translational Medicine, like. So there's translational medicine, translational research, translational science. It's all the bomb now, and it's these are just journals focused on it, and it takes place in industry, takes place in academia, and these are just mastheads of papers around doing drug discovery in academia, like a lot of you are doing, and a lot of you will be doing, and some of you may be doing. The other thing, because Rupa mentioned it so specifically around the Catalyst program, and the fact that the Catalyst program supports projects around new drugs, new diagnostic tests, new devices, new digital health. If you look at the bottom two rows, there's a big difference, and I know you all know this, but don't write these numbers down, even though I typed them down. There's huge, huge, huge difference in how long and how much money, and And drug discovery and development takes 15 years. And you know, an app, a cool digital app for the what's it called? The App Store, the Apple Store or the Android Store can happen pretty quickly. And then diagnostics and so I'm just pointing out this to you, and this is really relevant, is that when you saw those pipelines that Rupa was showing, they were all the same color. You know, they're shaped the same, they look the same, they had the same. But, you know, the one on drug discovery was like a 20-year thing, and the one on digital health was like a 3-year thing. I'm just pointing it out, but I know you all knew that. There's a lot of resources. I mentioned venture capital, you know about that. I mentioned, at the one at the top there is, yeah, wow, it's at the top. How coincidental. The Small Molecule Discovery Center, which I run. So that's a campus core facility. If you look in the middle, a lot of you, I'm not going to ask for a show of hands. A lot of you have heard of Y Combinator. A lot of you have heard of like Indie Bio Techstars, these accelerator programs that are just fantastic. The reason I refer to those as capital A accelerators is that if you get your project in there, and when Nate talks about NSF I-Core in a couple of weeks, you'll be hearing about this in spades. But the accelerator takes a little tiny piece of the pie, not that big, but it takes a piece of the pie. The venture capitalists, of course, they can take a pretty moderate piece of the pie or a pretty significant piece of the pie. The lowercase a accelerators are like Catalyst or Spark at Stanford or Ignite at UC San Diego. You get the thing. Catalyst, Spark, Ignite, you know, enthalpy, heat, fire. We don't take any piece of the pie. And so we help people like you accelerate your projects, but they're your projects and we don't get any help piece of the pie. And Nate will be talking about incubator systems and philanthropy and the like. Okay. If there's no question, let me look at my clock. Oh yeah, we're fine. Okay, I'm going to shift now to talking about the target product profile. And this is just a sort of an initial snapshot. This is almost sort of an aside that these things called TPPs or target product profiles, they mean different things to different people. And I'm going to start at the bottom. I know I threw you on that one. I'm going to start at the bottom. And this is why target product profiles are so important to Bill Gates, the World Health Organization, well, USAID in the old days, Rupa Ramamurthy, for example, because Rupa here used to work on malaria and tuberculosis. Okay, why it's so important is this, and we're not going to get into this, but it's analogous. Listen, this is important because it's analogous to what I said, that if you're developing a drug for teenagers, unless it's for a life-threatening disease, it has to be free of any cancer risk. If you're developing a drug for a very common, sporadic, very common disease, it sure as heck has to be a pill and cannot be an IV. This is like that. This is a third example, just like that. It goes like this. If you're developing a drug for malaria in the equatorial areas of the world, like in India or South America, or used to be China, you know, where they're developing nations with relatively low income compared to the developed world. We used to refer to it as the third world, and I know that we don't refer to it that anymore, but the developing nations, and either you all know this or it makes complete sense, is that it can't not be an expensive medicine. It cannot be an expensive medicine. So for example, one of the medicines here that's being worked on across the street that I'm looking out the window of, it costs $1 million a year for this drug that's being developed. It's an expensive drug. It's actually a cellular therapy, so it's not really a drug. $1 million a year, $1 million a year, and everyone's just ecstatic about it, including, the pay, Medicare, people that pay, or at least up to recently. But here's the thing. If you're developing a drug for malaria, not only can it not cost $1 million or $100,000 or $10,000 or $1,000 or $100, not even $10, it has to be, and Rupa will correct me right now if I get this wrong, it has to be on the order of $1, a single US dollar per patient per day, because if it's not $1 or less, it's not feasible. It's done. And so at the bottom bullet here, the WHO uses TPPs to sort of remind people like you and me and us that the economic considerations are tantamount and they can't be ignored. What I'm kind of talking about mostly is at the top bullet, a pharma company using these to basically manage a project and make decisions. Venture capital, oh, this is cool. Venture capitalists like them because it helps them decide what they want to invest with. And what's totally cool and is that Catalyst summer internship target product profiles have been used by professors and taken to venture capitalists to help talk them into funding their company. And when you talk to Sohaila or Nadarupa, you can ask about the specifics. And the FDA sort of uses them too. So here's the thing, and this is also sort of like slightly background, but I just want to hit these bullets. And it's sort of a long list, and I want to go through this. And the things that are underlined in red is sort of the key thing. And so what a TPP is, it really describes what features the product has to be successful. And I know you know what that means. Like I already said, it has to be oral. It can't be an IV. Or just think about, I'm just trying to think of an example at your house. I mean, this is true for anything. And just think how competitive, Amazon is online and why one product gets bought, another one doesn't get bought. So you really, it's the features that a product has to have in order to be successful. I mean, think about a computer doing artificial intelligence. If it has a GPU, it might be successful. It can't be successful with a CPU. Stuff like that. It's just like, what does it have to be to get purchased? And here's this next thing is important, sort of to everybody on the call, because it's sort of like anti-academic slightly. It's sort of like pro-industry, sort of. And you can see I put it in italics. It's beginning with the end goal in mind. And what do I mean about academia and industry? So when you're in academia and you're doing science and you're doing this and you're doing that, and you get a result that was unexpected, you're like, oh my God, if this repeats, I'm golden. This could be a Nature paper instead of a molecular cell biology paper. I'm going to pivot. And when you're in basic fundamental curiosity-driven research, you're taught to pivot. You're taught to follow the data. You're taught to meander. But when it comes to developing a product, when you're at a pharma company, biotech company, you gotta like the thoroughbreds. You have to wear blinders. You have to keep your eye on the fake rabbit. No, that's dog racing. You have to keep your eye on the goal. So it really embodies beginning with the end goal in mind. Okay. So a target product profile includes all the different attributes, all the different features, all the different things, whether they're medical, scientific, technical, clinical, business, market data. Okay. Fourth bullet. A little bit redundant. Yeah, okay, it's okay. This is just sort of redundant. It gives you a clear snapshot of what it's got to look like. And so yeah, you know, this fourth bullet is not much different than 1 and 3. I admit that. It's funny. I have an abridged version of the slide. Now I just realized why I made the abridged one, because this is the unabridged version. Okay, this fifth bullet is the most important. 5th out of 6. And I want you to think about this. It identifies the optimal and the minimal product attributes. And before I go to the italics, I just want to give you an example. And it's that example for the drug for obesity. I'm not going to say it for the 5th time, although I'm about to say it for the 5th time. It has to be a pill. It can't be a shot. And so that's minimal. It has to be a pill. But as you all know, some pills you take once a day. Some pills you take twice a day, BID. Some pills you take 3 times a day. And I think we all know once a day is the best by far. I don't know about any of you that have to take 2 pills in one day, but for some crazy ass reason, it's hard to remember to take you always remember to take the one in the morning. Maybe not you. You often forget to take the one at night. And so here's the thing. This is important. With respect to the scientist that has the drug, she's like, look, it has to be oral. And what's best is if it's 1 a day. If the pharmacokinetics and pharmacodynamics are such that it only takes 1 pill a day, that's the best. But if it has to be BID is twice a day, that's okay. And so just to be really clear then, the minimal product attribute would be it has to be oral. It has to be taken by mouth. Optimally, it'd be 1 a day, but twice a day would be acceptable. And so now we go to the italics. I can't read the part of them, but these will be the sort of the thresholds you use to determine if you should keep going forward. Because here's the thing. This is like super important. If she can't turn it into an oral drug and she convinces herself, well, a lot of drugs are injected, it's going to work. She's being really stupid, because she should stop then. Once you figure out, no, I can't crack this nut, I can't make it oral, you should stop. Because if you can't hit the minimal, it's never going to get sold, it's never going to get prescribed, it's never going to get used. The last bullet's obvious, is that, and I'll just use you all as an example, is that this summer you're going to be creating target product profiles for projects, and as of the end of the summer, it's going to be current at the state of the art. And the world, oh, you don't think the world's going to change this year, huh? So the world's changing, and it's going to change. And the thing is stuff can change. I mean, just think of these unbelievably powerful COVID-19 vaccines. The idea of RNA as a drug continues to blow my mind. And so something can happen that changes the landscape. And actually, it can change it the wrong way, right? It can change it the wrong way. That you could be working on something where an injectable route of administration is completely cool because it's life-threatening. Or just think of Ozempic and Mounjaro and Wegovy. Those little baby needles, that's not slowing anybody down from using it, I don't think. But I think many of you know that oral versions of GLP-1 analogs, oral versions of Ozempic, Mounjaro, and Wegovy, they're right around the corner. They're right around the corner. And so these needles for those weight loss drugs, and actually they're more than just weight loss, they're going away. And so the reason that your target product profile has to be dynamic is that when technologies change, things change. You might have been golden for 5 years, but now you're screwed. There's not going to be funding. It's not going to get approved. And so that's all obvious. Okay.
Hey, Charles, while you're on that last slide, we have a question from Peyton Skaggs. Peyton, the floor is yours.
Hey Charles, thank you for
Hi.
taking this question.
Sure.
My question is, do you think that it's like the strength of the TPP that can make the difference in getting from bench to bedside? Because when you start talking about like having to check all these boxes and meet certain standards, it seems to almost go from like the success of the product mattering less and rather like the way that it's presented. So
No. So listen, that's a cool question. Obviously, given what you said at the end, I wasn't being very clear. And so let me just tell you to your first question. No, it's not. What this is, it's a tool. It's just a tool. It's just an aid. It's to make things more efficient. And it's to identify the hurdles you have to surmount, the gates you have to get through to be successful. And it tells the entrepreneur, these are things I have to focus on because since this is going to be used in children, it can't be carcinogenic, and they can't forget that they have to do those tests. And so it's really just a tool. It's just, Rupa in a couple of weeks is going to talk about project management, capital P, capital M project management, which I know some of you know about. It's really interesting because it's more than you might think if you're not familiar with project management is. But project management has a lot of tools to facilitate projects advancing. They identify like rate limiting steps to make sure that, my God, that's like, oh, Jesus, this one, we can't do anything else until we get the decision made on this formulation, and everything else has to stop. Everything, if you're doing things in parallel. And so a project manager like Rupa makes sure the team knows that, and so they can put more effort on this one thing that will be a rate limiting step if in fact they don't surmount that hurdle or go through that gate. So it's really just a tool. It's really just a helper. Is that clear?
absolutely, thank you.
Cool.
Hey,
Okay.
Charles, do you want to take a couple more questions or do you want to keep going?
You know what? A couple more questions are fine.
Great. Anya, the floor is yours.
Thank you. I was just wondering, who actually determines each threshold in your TPP? So is it the team deciding where
Yeah.
some thresholds, or
Yeah,
is
you
everything
know.
FDA?
No. No, it'll be you and your friends on the team with the input of your catalyst directors and then your professor, and then because it's funny, Anya, I was thinking that I was clear. I mean, I use easy examples. Is it carcinogenic? Is it not? Is it oral or not? But you could have like really subtle examples like this. So listen to this. This one's cool. Our drug candidate has an affinity for its target of 1 micromolar. And that means that when we turn into a pill, it'll be the size of like one of those big. God, you know what's interesting about that? For those of you that know this, UCSF was behind a lot of the discoveries in HIV/AIDS. And when the first antiretrovirals came out in the late 80s, the people that took those drugs, they were taking like 37 pills a day, like 5 different times. And today they can take like 2 pills, even 1 pill. And so the thing is, it depends. So the thing is, if you have a drug that has very low potency, and so it'll require you to take two drugs, two big pills at once, and some people have trouble taking big pills. They can take little pills fine, they can't take big pills. And so that's why it's sort of a shade of gray, because things like it has to be oral, black and white. But things like how big can the pill be is a shade of gray. And so you people have to figure that out. I hope that was clear.
That is, thank you.
Sure enough. What was the next question?
Shrey, would you like to ask yours?
Yeah, sure thing. Thanks, Nathaniel. Professor Hart, thanks. I just had a simple question. I guess initially I positioned it as therapeutic specific, but in general with digital health as well, we've seen a lot of difficulty in anticipating willingness to buy. So if you look at therapeutics, right, how are we going to probably at the stage of developing the TPP understand, potential list prices, negotiations with PBMs, and with digital health, we've seen notoriously long sales cycles, right? 18 months or so without actually figuring out whether people will pay for a clinically efficacious product. So I'm wondering if you have any thoughts on that. Thank
Yeah,
you.
you know what?
Yeah.
I followed your question very closely. And think back to my thing about $1 million therapy for sickle cell disease and a $1 therapy for malaria. And you all know the connection between sickle cell disease and malaria. And so $1 million is fine for sickle cell disease in San Francisco, California, but the sister indication of malaria in sub-Saharan Africa, it's got to be less than $1. And so your points about reimbursement and payers is going to be a feature of a later on workshop, and it's a super important consideration, especially in the context of managed care and the current healthcare system. So yes, exactly that. And if you look through the template for the TPP, you'll see that the fields that you're alluding to are there, and there will be talks on it later. Okay, here's the thing, real quick. I just looked at my clock. It's gonna be no problem. I'm gonna be able to finish right on time. But since we only have 6 minutes, we have 8 minutes. Oh my God, I feel so much better. I'm gonna move ahead because I do. Okay, so here's the thing. You all go to the pharmacy. Oh, wait, Himani, I see you have your hand up. So Himani, what's your question?
Yeah, as new to this concept, I was wondering, even before this concept, this TPP, we always keep in mind before discovering the mutagenicity and tetragenicity. So does that mean this TPP is the new way of saying things we are already doing? Because
No. Wait.
Yeah.
Let me say one thing. Let me say something back, because I used to work on a drug for glioblastoma multiforme, GBM, primary brain cancer. Awful, just unbelievable disease. No. Mutagenicity and carcinogenicity absolutely do not come into play. There's not even, you don't even, like I said earlier, you don't even ask the question. You don't care. There's absolutely no relevance of carcinogenicity for a drug for primary brain cancer. So the specific questions asked and the hurdles and gates for each are different for every single product. And here's the thing, when Rupa showed that slide of a pipeline saying, oh yeah, this is how drugs are discovered. You identify the target, you find hits, you advance the hits to leads, you advance the leads to candidates, you put the candidates into clinical trials, they go from IND to NDA to the pharmacy. Oh wow, it's like one step in front of the other. There's a rubric for this. No, it's not true. That pipeline is just a visual aid. Every single drug in a pharmacy, every single drug in the pharmacy took a different path. Every TPP is different. Every drug discovery is different. Every digital health project's different. Every medical device project's different. So every project's different. And you don't want to confuse there's one size that fits all, with this idea that there are some common challenges, like your friend, I think Shrey just mentioned a second ago about managed care and decisions made by pharmacy benefit manager, PBMs and the like. Okay, is that okay? Imani, you can write me an email and elaborate on that too. Okay, here's the thing. You go to the pharmacy, you pick up your box, and there's that thin paper, that super thin paper inside that you can almost read. That thin paper is called, not surprisingly, it's called the package insert. Not surprisingly, it's the package insert. It's that super thin paper. You've all seen it. I'm not going to ask how many of you look at it. That was half serious, half joking. It's also called the product label. It's also called the prescribing information. Those three descriptions are synonyms. They're exactly the same thing. There's no difference. The product label is identical to the package insert, which is identical to the prescribing information. You've all seen it. This one's for Trulicity. This one's 26 pages. I just have the three there. They all have sorry, I always have to click back and say, they all have the exact same format. Either you know that or you don't. They all have the exact same format. And this is it. This is the format. They all have these 17 main things. They all have these exact same things. Now, my eye is looking over to the left where it says warnings and precautions, 5.4 heparin-induced thrombocytopenia. Well, obviously those are specific to this drug, but section 5 is the warnings and precautions. So like for me, those things in red that you see on the right, I mark those just for you, because personally, when I have to look at one of those thin pieces of paper, I don't want to say that I'm not interested in all these other things. That's not true. But what I'm personally most interested in, because I'm a pharmacologist and a biologist, is section 12. So I always go to section 12 because that's what, and actually it's really just section 12.1. My point is they're all exactly the same, which makes fantastic sense for doctors and pharmacists and nurses and PAs and this and that. Now, here's the thing. Catalyst really works on the left-hand side of Rupa's chart thing, pipeline thing, and not the right-hand side. And so I don't want to confuse anybody here, but I just want to be, and I don't think I will, but if I have, just let me know. On the right of this is about a target product profile. And really, that's about the approved drug. These things are all about the drug that's in the pharmacy, the drug that's in your veins, the drugs that's in your medicine cabinet and you're going to take tonight or tomorrow morning, or maybe you don't have to take. These are drugs. These are approved drugs. Catalyst, you all, and this is analogous for the other things, kind of, sort of, you're really focused on the left side of this, the discovery side, the preclinical side, trying to come up with a candidate. And so so the people here working on drug discovery projects, and I know there aren't that many, what you're really putting together this summer is a target candidate profile, It's what do the features of the candidate, the thing that's going to go into clinical trials, what do they look like? And so those are things like this. And Shrey, you'll see that the second from the bottom, yeah, I see you're nodding your head there. You're still on my screen. Great. And so I'm not going to read through these, but you'll see what I did with the black bold and the gray non-bold is the ones at the top and the bottom are going to be the same, whether you're a digital health solution, medical device, a diagnostic, a therapeutic. They're going to be the same. What's the competitive landscape? What's the FDA going to say? Can you make money? But then the ones in the middle that are in gray and not bold, those are the things that are specific. And so just look over to the left. That's me. What I did for 30 years was cell-based assays in test tubes, or mouse, rodent assays. And then if you're working on medical devices like Rupa is an expert on, you're worried about things like functional specifications, which I thought I knew what it was. Environmental issues. Okay, I just want to be really clear. I'm just making that point. The reason we call it a TPP, if someone says, and I don't want to question, well, why don't you call it a target candidate profile? Is because everyone, well, maybe not everyone, a lot of people have heard of TPPs. No one's heard of a TCP. And in fact, I might have made it up. Okay, here's the thing though. And this is serious. That wasn't not serious, but this is serious. You look at the top of this slide, and this is especially true for people in digital health and devices. And this goes back to when I answered the first person, about this just being a tool. And you'll see how you put project and management and capitalization, capital P project, capital M management. There's other things that are just like, almost like, sort of like TPPs. And you'll see them there, those three I list. I'm not going to go through them, but an MVP. And different people, different companies, different people have different names. And at the bottom, I'm not going to talk about these at all. But I told you in the beginning, I'm looking at my watch. I'm going to finish up right now. I told you in the beginning, twice when I was in Big Pharma, I was involved in moving a drug candidate into development. And when Big Pharma pulls the trigger, again, I mentioned the trigger, when Big Pharma decides, hey, we're going to start spending the big bucks, they usually create 3 documents. And they put together a team, an ad hoc team, a team that was brought together for this one purpose. So twice I was asked to join a target product profile team. It was very educational. But it's the same true with a CDP, a clinical development plan, and it's exactly the same true with the risk management plan and the so-called risk register. Those 3 things, the TPP, the CDP, and the risk register, are key documents that are often created in parallel. And when Rupa talks in a couple of weeks, she'll cover those. And so as Rupa already said, and Nate alluded to, you're going to do these sort of in order through the summer. These are examples of the medical field, and you can see on the left is sort of like the name of the field. And on the right in italics is just, what does that mean? What is the current standard of care? And it kind of makes sense. That's how people currently take care of obesity or teenager hypertension or primary brain cancers. And so in terms of you all getting help for filling out the medical fields, you'll get access to the application from the professor, and as well as her or his finals presentation. And that could have some good info. Nate will be providing, or maybe he already has, we've got a really, really nice spreadsheet. Excuse me. Oh, I guess it is a spreadsheet. I apologize. Yeah, it's an Excel. But it has hyperlinks, all these great, I know no one can read this. I can barely see that some of it's in blue and some of it's in black. We have all these totally cool websites. Almost all of them are open. There might be a couple that need a password, and then talk to Sohaila, Nate, or Rupa. I already talked about minimal and optimal, and you'll see, like I said, and I'll just use that top one here. And as I said, a 1 micromolar potency would be great, but the pills are going to be really big. And so an optimal would be, 10 nanomolar potency, 100 times more potent. Minimal 1 micromolar, optimal 10 nanomolar, stuff like that, whatever. And same with animal experiments. If you look on the right here, and I've already mentioned the top two in spades, and this bottom one goes to Shrey's point is, well, how much is it going to cost to make this thing, whether it's a drug or a chip or an app or a pill? I'm definitely skipping this. I had to, you know what? Yeah, maybe maybe Rupa invite me back. I'm gonna stop right now.
Of course. Thanks a lot, Charles. And I just want to cover one thing which I forgot to cover, confidentiality. All of y'all have signed CDAs or are covered by the UCSF policy. What this means is you do not discuss the project with anyone outside. Don't post on Facebook or whatever other platforms y'all use. What y'all can speak about is, I had a great experience, or I learned. The lectures, you can talk about the lectures, but not about the projects. And even the lectures, whatever slides you get, keep those with you. Don't just distribute the slides. Definitely all the project information to be kept totally confidential. That's one thing I wanted to mention. Then if you all have any questions for Charles on the TPP.
Yeah, sorry I went 5 minutes over. So
That was great,
we don't
Charles.
have to ask.
Thank
Yeah,
you.
thank you very much. We don't. You don't. You can text. Write me an email if you have any questions, or come by and see me. And it's great meeting you all. And I'll probably see you at one of these upcoming workshops as well. And so, best of luck this summer and with the other stuff you've got going on. Okay.
Okay, thank you, everyone. Thank you, Charles. And Nate, Sohaila, or I will be working with y'all and the teams. So look forward to meeting y'all. Nate, any closing words?
Nope. All good. Thanks, everyone. Look forward to the development of the TPPs. Thank you for your participation. Feel free to reach out with any questions.
Okay, bye, everyone.